you to the Volterra lecture of the Center of Digital Life, Norway. And we are particularly happy that we can welcome Professor Bernhard Palsson to give his inaugural Volterra lecture. As you may know, the Volterra lecture is a national lecture series where we invite distinguished scientists that are relevant, doing relevant research for the digital life projects. Bernhard is the Galetti Professor, is also here, of Bioengineering, Professor of Pediatrics, and Principal Investigator of Systems Biology Research Group in the Department of Bioengineering at the University of California, San Diego. And he's also director of the Novo Nordisk Foundation for Biosustainability in Copenhagen, Denmark. I think that I'm not going to say much more, Bergen. I think that the title is here, An Emerging Era of Genome-SCALE Science. We are looking forward to hear your talk. The floor is yours, please.
Is this working? Yeah. So you can roll the recording, I guess, then. I'd like to start out by thanking you for inviting me to come here to give this talk. It's been a fascinating visit for me already. I am actually born and raised in Iceland. I left Iceland when I was 20 years old, so I never knew what it was like to be an adult there, but I was taught that Icelanders are descendants of Norwegians. Immigrated the country was mostly settled by just people from the western part of Norway. Although now that we have full genetic definition of the population there are more Celtic mitochondria there than history records, and probably also some people from the northern part of Norway that are found in Iceland. But I was checked into a hotel called Bergen, and I realized that my middle name is Ern, so I decided to put it in full here, which I normally never do. Now, the title of the talk is basically derived from the question that Jon Olav asked about the term systems biology. I think of systems biology much more as a genome-scale science. It arose after the first genome sequences came out. It came into being as a field. It really started out as being molecular systems biology, but it's now growing up the biological hierarchy and beginning to be able to analyze more and more complex biological functions. So I think of systems biology as a genome-scale science that is enabled by having genome sequences available. So today I was not going to go into much detail on any particular topic, particularly mathematics, but try to give you a balanced view of experimental biology as well as data analysis and data modeling. I will say that I was quite stimulated by the first two talks here this morning because FBA actually originates from the problem of parameter uncertainty. And as Eivind said, showed us, and we saw in the first talk as well, that the kinetic parameters in particular are pesky and difficult to deal with, and now that we understand better the outbred nature of populations, they will always be variable. We will never know them very well. And this was a problem back in the late 80s when I was starting to build my career as an assistant professor. We were always looking at this equation of these dynamic mass balances that we know is fundamental and describes basically fundamental functions of life. And we could factor it into this matrix, stoichiometric matrix, and the vector of reaction rates. And we were looking at it, and darn, it's so hard to get all these numbers for these reaction rates. I mean, how are we ever going to get that? But look at that S. You know, that S is nice. You know, it's knowable. You know, stoichiometric coefficients are integers, and if you can get S defined on scale, what can we learn from it? And that simple question, what can we learn from S, was actually the origin of flux balance analysis. And not to get into it now, but the notion of steady state can be discussed from a mathematical standpoint. but perhaps more importantly from a biological standpoint, as a homeostatic state. Once an organism finds the right set of kinetic parameters to operate and survive, it keeps that steady state. It will not take large steps away from that in parameter space because that can mean death. So it's really that homeostatic state biologically is quite related to the notion of a steady state mathematically. That state is very fundamental to all of biology, although it may not be strictly, true under all conditions. So, and variations around it do need to be studied. So let's talk a little bit generally first about the bottom-up approach to metabolic systems biology. This is the mechanistic approach based on genetics, genomics, biochemistry, thermodynamics, and the like. And of course, there is the complementary top-down approach with omics data, which I will also talk about later on. I just want to talk a little bit about available reconstructions, what they tell us, and how the content is growing with time. And I wanted to say a few things about human metabolism because there's a lot of interest in cod and salmon here. And as was already mentioned this morning, I believe by Jon, that there's no reason why similar reconstructions cannot be built now for these two very important species. And we'll talk a little bit about how to describe mechanisms beyond metabolism. As you know, proteostasis has become quite important recently, the proteome allocation, how is space allocated inside a cell, and also the degradative mechanisms or damaging mechanisms on the proteome, particularly ROS, as the stress has become now into focus, and we can describe that mechanistically and put that into these large-scale constraint-based models. I picked out a few application examples rather than stay with mathematics and talk first about proximal causation in biology. And that's, of course, the way biological organisms respond immediately to stimuli without any change in the genetic background. And then maybe more importantly, talk about distal causation in biology, which is the causation we think of as evolution. How do things change in an organism over multiple generations as the genetic material can change? This is what's described by the objective function in constraint-based analysis, and I just will say right away that the objective function is the biologist function. It doesn't exist in chemistry and physics. It's specific to biology, and it does describe biological causation that we think of as distal causation. And it's quite an interesting function, and will continue to be interesting. And I will just show you that we can now address it experimentally in the laboratory and at least learn how microorganisms do respond to the imposition of different objective functions. Quite an exciting new line of biological inquiry. So let's go through a broad introduction here to begin with. This is a slide that I put together some years ago, ended up in Nature Biotech in 2000. I was witnessing more and more sequencing, microarrays, people walking around claiming we could measure 1,000 metabolites, which I thought was just a dream at that time. I started out as an analytical chemist, and I knew how difficult it was to measure one molecule, let alone hundreds or thousands simultaneously. But technology progresses, and we of course now can molecularly profile cells in great detail. And this was already clear in the late 90s, so at the end of the 20th century. And the need for integrative analysis came up, first through bioinformatics, sequence analysis, and then more through mathematical models of complicated integrated biological phenomena at the molecular level. And that's kind of where the COBRA tools came from. Now, it was also mentioned this morning, I believe also by Jon Olaf, that biology will more and more become systems biology because biology is all about systems. It's fun to know what the protein does or a gene encodes for, what the metabolite does, but ultimately it has to fit in the context of the rest of a system and not perturb its homeostatic state, do its job and so forth. And this also is an older slide that I used to illustrate what GENOME-SCALE viewpoint means. Here's a simple pathway that you might be interested in, looking at allosteric regulation or cofactor use or whatever. But in reality, of course, this is a part of a network, and all the environments around that factor in the network, around the pathway in that network are very, very important. And now that people are doing metabolic engineering, they are finding out more and more that the host factors the factors that characterize the environment of a synthetic construct are actually quite dominant. This is an early reconstruction for a thermophile. No, this is for a methanogen. And the beauty about these reconstructions that we realized early on is that every event in there is tied to a gene, genomic location, promoter, regulators, and so forth. And these genome-scale networks became a way to study genetics in great detail, which was a bit of a surprise to many, and also the fact that metabolism is so variable from one organism to the next. Thermophiles, for instance, synthesize amino acids quite differently than organisms growing at, say, lower temperatures. So this gave a rise to network reconstruction. Okay, this is a very laborious process, but it has to be done if you want to build meaningful relationships in biology. A well-curated network reconstructions allows you to relate myriad of variables and create a coherent whole. Sometimes they work well, sometimes they don't work well. There can be gaps in them, they can be incomplete, they may have errors in them and so forth. So the key here is to have a quality controlled process to understand what you actually have in the end. And this is a workflow. It's written as a pipe, a whole kind of data types going in there, different mathematical methods, different QC, QA steps to make sure that you actually know what you have. And I'm always reminded of Manfred Eichen's quote when I look at that. So he said the hypothesis can either be true or false, but models have a third choice. They can be irrelevant. For instance, a reconstruction that is not elementally balanced is physically irrelevant. It can be mathematically accurate and all of that, but it may not describe reality for you. So we have digital life now. We have real life. And we know a lot about real life, and we can represent that in a reconstruction, which is a knowledge base that may include, like I said, genetics, genomics, biochemistry, and now increasingly protein structures, metabolite structures, cellular locations, and the like, that you can actually put into a knowledge base. And a knowledge base is very different than a database in that it grows over time in value, because as you curate it and add to it, it becomes more and more valuable. Whereas typically the value of data set decays with time. There's new technology that comes along, or what have you. The SOPs are different from lab to lab and so forth, but the knowledge base, the reconstruction is what remains. And then, of course, with some of the mathematical methods that Eivind mentioned this morning, you can take that knowledge base and mathematically represent it. There's really no theory in it yet until you start stating assumptions, but you can mathematically represent that knowledge base. And therefore it becomes queryable. You can just ask it questions, and then you can state hypotheses and build models of it, of course, too. I used to think of this as in silico cells, but now I'm taking liking to the term digital life. That's digital life right there. And I'm not going to go through this slide because Eivind did. There are two simple assumptions involved in here. Biologists like to think about genes and regulators and alike as, okay, I hit a constraint. I don't know, is this, oh, there you go. I think it's just, I touched this disconnected. But in reality, when you look inside the cell, nothing is independent of everything else that happens inside the cell, and that's captured in constraints that can be fundamental or data-based, but you still never can really calculate exactly what the cell does, and it is trying to satisfy survival criteria that we can restate as objectives, but it always has choices. You can always migrate parameter values, and that's what's supposedly captured by a good solution space. So these are very simple fundamental concepts, and like Eivind said, a high school kid can really download the Cobra toolbox and start to calculate. So what are the resources? This book here on the title Systems Biology, that again should maybe have the molecular systems biology. So this is a textbook written for a class that we give at UCSD. There are now conferences. There's a conference series on Cobra methods. And by the way, COBRA stands for Constraints-Based Reconstruction and Analysis. And FBA is one of the COBRA methods that's available. There are now probably a couple hundred of them. And as we saw this morning, the list is growing. So these conferences are held every 18 months. I think the next one will probably be a year from now in Seattle. There are primers available. The 2010 primer in Nature Biotech is 2 pages. That's where the image came from on the previous slide. And there's a supplement with it of examples of how to do COBRA calculations. And people that sit down, read that, download the COBRA toolbox, they're actually calculating biologically meaningful things within a couple of hours. So the barrier to entry into this field is actually quite low. And there's a more detailed primer in Cell a couple of years ago. We have now a collection of the reconstructions of the models, the ones that have passed certain QC/QA tests, and we are going to try to get as many of them out of the literature now, go through these QC/QA tests, and hopefully work with the authors to get some unified standard across the whole field to get a large number of these models. Computational resources, there are the 2 COBRA toolboxes published in Nature Protocols in 2 popular papers, and now of course there's COBRA Pi, and there's a number of other ways to become computationally operational with COBRA methods. Visualization tools have been a problem. The best network visualizer was written in a piece of software called Symphony, for simulating phenotypes by Genomatic, a startup company I put together with some others some years ago. That never was available publicly, unfortunately, but now Zach King has put Escher out, which is a rip-off of that software, and it works very well. A lot of people are using it. There's a rapidly expanding user base of it because you can make beautiful network maps very quickly. The Protein Data Bank has adopted it, for instance, and so have others. And finally, every lecture from that class and that textbook is now available on YouTube. So if you want to sit down and listen to me for hours and hours after today, you can. Okay, so those are some introductory comments. Let's talk a little bit about available reconstructions. So E. coli, always a key model organism. That's true in systems biology too. I'll talk a little bit ago about going beyond metabolism, as I mentioned before, to look at the proteome. And now we have structures, protein structures embedded in reconstructions, and Recon 3D of the human network is going to have that information in it. And I'm putting that in here to anticipate your desire to have something similar for cod and salmon. And the need for visualization, I probably will skip that. So, COBRA methods work 1, 2, 3 for bacteria, and this, of course, has many applications as I'll talk about. I like to put this image up from David Goodsell because it illustrates the magnitude of the problem. It's a multi-scale problem, of course, going from small molecules to the bigger ones. E. coli has about 2.5 million individual protein molecules, and that's true of most enterobacteria. So some elongation factors, the main elongation factors require 200,000 copy in a fast-growing cell. So it's almost 10% of the proteome in one type of a protein. So which means that everything else has to go way down. There are about maybe 2,000 proteins, say, expressed under a given condition. So let's say it's 2,500, divide that into 2.5 million, you cannot have no more than 1,000 copies of each on average. So if one is going to take 200,000, a bunch have to go into much lower concentration. So that represents constraints. How do you allocate the proteome, as an example? We can now profile, of course, the transcriptome and a number of other things. So this is actually a defined problem in so many different ways, in molecular details, in genetic terms, in survival criteria, and so forth. And therefore, molecular systems biology has been very successful with bacteria. This is the history of the E. coli reconstruction, starting in the 90s. Building a reconstruction is not easy, it's very laborious, a lot of checks and balances. And you can see here over time how particularly the gene count has gone up and the number of reactions. And if you have good eyes, you can scan up this list because it describes how the different cellular functions were layered into this reconstruction one after another. And of course, this took a big increase when the first genome sequence came out for E. coli in 1997. And you see two names at the end there, Colton Lloyd and Jonathan Monk. They put together the new reconstruction that has 1,515 genes in it, and that will be printed in Nature Biotech this month. And that's actually quite a lot. I mean, there are 4,500 genes in E. coli, about 1,500 of them are white genes with very poor annotations, but there are 3,000 genes in E. coli that are well annotated. Half of them are already in here, and a big chunk of the other half is now in the proteostasis models that I'll talk to you a little bit about. Now, let's talk about then some biology here. So this is MG1655 is a particular strain of E. coli that's a laboratory strain, like I said, with 4,500 genes in it. It's a genome-reduced strain compared to most wild-type E. coli cells strains. So it's more like 5,000 to 5,500 genes. Now we have genome streaming at us for different bacterial species in mass. They're just coming in droves, and we are now in a position to deal with that. We can map these well-curated reconstructions onto new genomes, and we can do very valuable things when we do that. And we first realized that in this paper published in PNAS in 2013. Let's see, okay, there it is. When we could look at 55 sequenced E. coli strains and map the MG1655 reconstruction onto them and study them without doing a single experiment. And we studied the pan-genome, these genes that are variable from species to species, and the biggest diversity was actually in the catabolic capabilities. E. coli can grow on many different substrates, and so there was diversity in that, and there was diversity also in transporters. And the upshot of this was that you could predict the colonization site of E. coli, because the nutritional environment in the body is quite different. And UTIs, urinary tract infections, of course, is a very particular environment, but then there is interintestinal, extraintestinal, and so forth. And this classification tree there shows that there are few metabolites that can be used as classifiers. Whether a cell is able to use them or not can be predictive of their colonization niche. And what surprised me even more is that it predicted oxotrophies quite well. We thought these were gaps in the reconstructions in the beginning, but they were real because genes were missing. And now we know that the oxotrophies amongst members of microbiomes are very, very common. And as I mentioned in our conversation during the break, folate, niacin, and thiamine, which are B1, 3, and 7, and tryptophan are very commonly oxotrophic between microbes and hosts. We now have thousands of sequences of E. coli, so we can now be looking at thousands of models of E. coli, and that leads us to a study like this one. This is from this paper that's coming out here in a few days. There is a core metabolic network that's common to all strains of E. coli, so we have a good model of that, so we can understand what's common between all of them. There are other genes that are also found in all of them, and then there's a set of variable genes. This is the pan-genome that actually gives a strain its unique characteristics. This here could maybe be used to define E. coli as a species, because that's what's in common between all of them. We now have protein structures on there, so we can actually now look at every open reading frame and look at the sequence variations in them and see which ones are highly conserved and which ones seem to be variable across the species. So HISD, which is an early gene in histidine biosynthesis, is the one that has the biggest variation for some reason. And we are now looking at what might the possible reasons for that be. So this is going to lead us to all kind of new hypotheses and studies that we can do by querying a reconstruction like this one. There are some compelling applications beginning to show up, having a set of models like this. We are now looking at sequences from fresh clinical isolates and actually predicting pathogen properties just based on sequence. We're also doing something that's even more interesting. We can now get deep sequencing of stool samples, and in these papers we show how your E. coli strain can be reconstructed from your stool sample. And we're actually doing this for patients now that have IBD, various forms of inflammatory bowel syndrome diseases. And some of those E. coli strains are quite interesting, and I would certainly not want some of them in my gut. So the number of applications that is now exploding, based on these reconstructions, are going to be quite exciting to deal with in the coming years. So the statement that was made this morning by Jon that all biologists will become systems biologists, well, now you can start to see why. You can study diversity, you can study infectious disease, you can study structural biology in the context of natural evolutionary outcomes these strains came from, strains came from. So let me show you one tantalizing opportunity that is now emerging and will have to be addressed within the next few years. And that is the possibility that we can now sequence virtually every systemic infection that is going to be diagnosed in a patient. We're going to be able to build all these models, personalized models of your pathogen now. We can just do it over the next few years. So in the United States, here are some organisms most commonly diagnosed in the hospitals when people have systemic infections. So there we have Pseudomonas aeruginosa, Staph aureus, and of course MRSA being a problem, and then Salmonella and E. coli being the most common. And what you see up there are the annual caseloads in the United States for these organisms. Systemic infections treated in a hospital, of course not all of them. Here's a graph over time of the number of sequences available for these strains, and this has been growing exponentially. That is a log scale. And if you extrapolate these to intercept the caseloads and ask yourself, at what year are we going to have the sequence of every single pathogen potentially diagnosed and isolated in the United States? Well, it's in 4 years. So this is coming fast and furious. So this is something we have to deal with. And this is a problem where the type of systems biology we've been talking about here today will have a big impact. And I'm pushing a project now called PASTU-21, which is going to be an IT-type approach to monitoring, managing, and potentially treating infectious disease on scale in the 21st century. So that's a little summary of metabolic reconstructions and where they're going and what the future may look like. I'd like to spend a few minutes now to talk about how these models are growing in scope. So here's the metabolic network. Here's a reaction which of course is to be catalyzed by a protein and maybe a protein complex that may have a prosthetic group in it. So this has to be synthesized by the cell for this reaction to happen. And we can trace back all the reactions that actually make that complex all the way to the genome sequence through transcription, through translation and transcription. And each one of these steps is going to demand nucleotides and amino acids. And we can actually form now a fully integrated network of the metabolism and protein synthesis. These are called me models for metabolism and expression. And they can now compute up to 80% of the proteome by mass in a fast-growing cell. So here are some of the attributes of these models. They have this transcription and translation built into them. We even have now the protein localization pathways that localize the protein. We have the structures. And then this enables a variety of applications. Shockingly, prediction of temperature sensitivity of growth. This I'll show you more about in a moment. So these are me models that people are building now for a variety of prokaryotes. And there are even brave people that want to do this for eukaryotes. That's the luck, because it took a while to reconstruct this. As long as you can reconstruct the networks, you can do it. So we are now, this is for metabolism and protein synthesis, and we are now growing this to look at other functions in the cell. And this is the proteostatic network in E. coli that has proteases in it, chaperones and alike. And Kachem has built a reconstruction of that. And it turns out that many of these processes here, like folding, aggregation, kinetics, and so forth, can be described as a function of temperature. And when you put that into one of these me models and then just dial in temperature and optimize the growth rate, you calculate this curve that is shown here. And there are some variations on it here, nutritional variations. And here's independently obtained data sets from the literature. You map this on one another, and this is just shocking how well this happens. And you can divide this into 3 regions here. The Arrhenius region, where the growth is growing with temperature. There's the optimal, the typical physiological state, and then the stress condition when you go past the optimal temperature. And this, these graphs down here, this is coming out in PNAS in just a few days, show how this, the allocation to protein synthesis, chaperones, and metabolism is predicted to be as a function of temperature. We know about the ribosomes and translation, how that changes with growth rates, so the model calculates that. Here is the allocation to chaperones. Here are the lower temperatures. and when the growth rate goes up and hits that maximum temperature, all of a sudden, as you increase the temperature, this curve turns around because bigger and bigger fraction of the proteome is now found in chaperones to fold the protein correctly, and there are fewer and fewer protein available to drive growth. So you can just calculate this trade-off between proteome allocation to the growth processes and protein maintenance processes, say. We're doing the same thing now with ROS stress, and we have a lot of data that is supporting this. We can now describe the damage mechanisms associated with ROS stress, of which there are a few. The one that I found the most interesting was mismethylation. So ROS species can oxidize iron in an enzyme to go from a +2 state to a +3 state, and it's dislodged. And a zinc or a copper divalent cation can go in there, and the cell can't get rid of it. It's stuck in there. This enzyme is still be functional, maybe at 1% of the Dmax, and the cell has lost 99% of its investment in that particular protein. The macrophages, when they put pathogen into a phagosome, douse it with ROS and zinc at the same time. And zinc goes into various proteins and weakens the pathogen substantially. So we are using this to do predictions, the straight trade-off of growth versus being able to deal with oxidative species, kind of like we did the temperature earlier with chaperones, and we are predicting how the metabolic fluxes have to change, and we're now evolving E. coli in the lab to look at if the mutations that happen correspond to those predictions. So this almost seems like science fiction, that you're able to do this on a genome scale, but this is happening. So I think we're going to have all the major stress functions that prokaryotes experience soon in a computational form, and you can study them in the context of everything else that's in the cell. Everything else being what's in the me model. Not quite everything else, but everything else we can account for. So that's what's ahead for reconstructions and computational biology of prokaryotes. Let me say a couple of things about eukaryotes. So this is Recon 1 that came out in 2007. Build 35 of the human genome was done in 2005, and there was fairly good annotation then, appearing of euchromatin. This is when we got downgraded as a species from 100,000 to 70,000 and finally to 25,000 or so genes. And Recon 1 was built as a multi-scale knowledge base, as pictured here. This took a lot of work on the behalf of many people, and that's the nature of getting, that's the nature of the reconstruction process. It's very laborious, and it's good to multiplex that and subtask that. The new reconstruction called Recon 3D accounts for the fact that protein structures and their availability has been exploding over time, as shown on this diagram over here. And we now have a reconstruction that will hopefully be published by the end of the year of human metabolism that accounts for over 3,000 genes, a lot of reactions, a lot of structures, and so forth. I thought this very interesting, that there are 19,000 protein are now functionally annotated in humans. There's probably more than 20,000 genes, but 19,000 are annotated, and we have structures for most of them, but many of them, maybe not most of them. And metabolism is actually 17% of that. That's actually quite surprising that the metabolic network is so important to human life. I mean, in terms of gene count. We thought that there would be relatively few of them. So this is just a snapshot to show you what reconstructions of human metabolism might look like, or cod metabolism and the liver potentially in the future. We just don't have only the pathways, but you have the protein structures. You can calculate the abundance of proteins needed based on their activities and so forth. And we also have full mold files and so forth now of the metabolites, so you can start to look at the structures of the metabolites. So metabolites that are not natural, various toxins and so forth, can now be studied also from this standpoint to see where they are likely to be metabolized or where they're likely to maybe interfere with the intrinsic metabolic machinery. And you can use this also to study conserved moieties metabolism for those of you interested in finding a good basis for the left null space of this documented matrix. Okay, so that's what's happening in reconstruction. So we are entering a new world in terms of data types presented and integrated, and the scale of this is done not just for single organisms, but across the phylogenetic tree. And again, quoting Young, every biologist is eventually become a systems biologist. You can see where this is going. So this is very fundamental to all biology going forward. So I figure I've used what, 20 minutes now or so? Or what? A little bit more, 22? Yeah. Now I'm going to go through examples, and I'm sensing that I may have less time than I thought. And let's go through one good example of how systems biology is used in practice. And we're going to be mapping metabolomics data here because I thought that was of interest, and also look at challenging the steady state assumption as an example. So we've been looking at blood storage. Red cells are put into bags and kept for up to 40 days during a time over which they decay. We can now collect as a function of time various measurements, and they're shown here. So the exometabolome, the media, the endometabolome, the inside, and various other measurements done in a blood bank, pH, PO2, and ions, and so forth. And we can map this now onto a metabolic map that we have available of the human red blood cell. This is generated by taking Recon2 and mapping deep coverage proteomic data sets onto it to see which protein are found. And you generate a map like this, and it's fully functional. There's complete pathways. It represents a lot of the cell's physiology. Not likely to happen for every human cell types, but it's happening for red blood cells. It's happening for platelets, and who knows how far this can go. This is what you see in hematology textbooks, the basic pathways. You always get very complicated lipid metabolism when you start doing this. But so we have, let's call this clinical data. That's omics data, and that's a reconstruction. So we can combine these now to study the metabolic decay in red cells during storage and how the lesion forms. And you can start that by doing big data type analytics. This is a PCA plot of this data set. And you can see that it breaks the decay path down into three distinct states. There are metabolism in red cells does not just decay exponentially to oblivion, it shifts gears. So there are three distinct metabolic states in there. And you can see some of them very clearly. This is adenine being depleted in the medium. This is hypoxanthine building up. You see here is ATP going up and then going down. And you can see that the shifts in these metabolic profiles correspond to these kinks that I've shown there. And this is for platelets, where there's one kink. I'm not going to say more about platelets. So as biologists, we want to perturb this. Since we said, well, adenine must be a problem because it is depleted. You boost it in there, and there's no change. It just goes down to a certain level, and then it's not touched. There's something else that blocks its uptake. We looked at different sugars because there were other sugars in blood, mannose and fructose. That doesn't change this basic pattern in the PCA. Then we wanted to speed things up because things happen very slowly at 4 degrees, and asked ourselves, can we do this at a different temperature, do the experiments faster? And it turns out that this basic decay pattern scales with temperature. If you do that PCA plot at different temperatures, it still kind of looks the same way, which is a little surprising, was a surprising result. So we perturbed and not really yet has had a big success in changing this fundamentally. So this metabolome and its decay is a fairly constrained process. You know, it's happening the same way over and over again. Now, we can use this to define biomarkers, and this is maybe one of the more valuable outcomes of research like this. We found basically 8 biomarkers that are able to distinguish these three phases, and it turns out that this one here, hypoxanthine, is probably the most informative one with respect to clinical implications. Mouse data shows that the more hypoxanthine has built up, the less the cells function upon transfusion. Not been shown in humans yet, we'll see that. But you can get biomarkers, and then based on those biomarkers, you can now build statistical models, and that's shown here. So here is the core metabolic network. In red here are the biomarkers, these different points that you may want to monitor, And these call-out graphs here show a particular metabolite that is just calculated based on these other biomarkers, based on after the statistical training. And you see that the dimensionality, the flexibility in this metabolome is limited. It can actually be described by only 5 biomarkers. Too much text. Now, but we can now get to some mechanistic hypotheses based on this, and let me just show you a couple. Surprisingly, there's a high level of malate in red cells. There's no TCA cycle there, but there's still a lot of malate in the millimolar range, and this was a surprise. And it's related to citrate that is sometimes used in the buffer that their cells are extracted in. And we used the C13 labeled of citrate to confirm the map, basically, the predictions of fluxes. Then there's another quantitative prediction here. It's a little more interesting, and it relates to that ATP decay. So this is the 2,3-DPG that is unique to red cells, high concentrations, binds to hemoglobin and controls its oxygen affinity. Was always assumed to just decay by dephosphorylation and then go through lower glycolysis. And really balance all the equations, including pH, the hypothesis that this actually goes back this way. This mutase actually does become reversible. It goes through this ATP generating step in addition to pyruvate kinase. So you get twice as much ATP for every one of these molecules. And this seems to be what really drives that first healthy metabolic state that you see there. And this is what we call unstable FBA. So not a steady state, and that's when you actually look at each one of these concentrations in the endometabolome that are decaying, and instead of putting S dot B equal to 0 for that metabolite, you put it equal to the measured slope here, and the error of it. So now you don't have it equal to 0, but you have it equal to data. So this is the way you add data into a reconstruction to constrain it. Now you get actually a flux cone that does not correspond to a steady state. It corresponds to that drifting state that you actually measure. So this is leading to a paradigm shift in this field, where systems biology is playing a central role, where you do your experiments, you do the statistical analysis, you do the mechanistic analysis, and now we are beginning to do model-based experimental work. So that's an easy to understand example. I hope I didn't go through that too quickly. But this is how you can apply bottom-up systems biology with omics data sets efficiently to learn a lot. Now let's see, how am I actually doing on time?
Well, you have 15 minutes
Okay.
for lunch.
Okay, so I will have to skip an example or two. Talk for too long about Icelanders being Norwegians. So we have been mistreating students in our lab. We've been fasting them and starving them and bleeding them on Sunday mornings and only taking 10 cc's or so. We measured their fasting plasma metabolome, shown here, some measurements, and the endometabolome. Inside, or is it the other way around? This is the endometabolome and the exometabolome in them, and then we put that, and because the fluxes in and out are known, and the flux might be reasonably well known, we can ratio the concentrations to the fluxes and get the pseudo-elementary rate constants that we call perks. That will be now individualized. They are different from person to person. And it turns out that then we genotype them all. And it actually turns out that these perks are more representative of the genotype than the individual concentrations. Now that we're talking about metabolomes and how they are tied together and so forth, these perks tie it together. They turn out to be better correlates with genetic parameters than the individual concentrations. So looking at the system rather than the individual measurements. And then you can simulate, you can build individual models for these individuals, and you can predict things like toxicity to a drug that was known to affect about 8% of recipients is no longer on the market and cause hemolysis. And we studied that a little bit further, but since I don't have a lot of time, I should say that 2 out of 24, or 8%, is approximately the number reported for a larger population. Obviously, some luck. You need probably to a few hundred to get at this. And they are predicted not to be able to respond to ribavirin. We couldn't get the drug, and we couldn't replete the students, so it graduated. to confirm these predictions. But ribavariant sucks up basically it's similar to ATP. It sucks up the phosphate as a dead end in a network and drains the cell of energy. But then we could do big data analytics of these different models, and we could get classifiers. And it actually turns out that this ribavariant sensitivity is related to those individuals that have higher activity and lower glycolysis, as well as the ability to move phosphate onto adenine. So those were the predictions here, and you can look at this as personalized medicine. Now, just thinking about time here. Let me just skip this example. We have looked at the all the data. These are 3 slides. We looked at all the data available for the NCI panel, NCI 60 panel of cancer cell lines, and analyzed all the data that's available for them. And basically, well, maybe it's hard to skip something like this. And we've been able to kind of define the hallmark of cancer metabolism. So glucose to pyruvate to lactate, known as the Warburg effect, of course, shows up very quickly, shown here, across all the data sets, all the cell lines. Less known is the ability, the desire of cancer cells to consume too much glutamine and secreted as glutamic acid or alanine. That's shown here. And a third one to pick up too much serine and export it as glycine, which is maybe not the strongest of these correlations. And when you look at the higher this overflow metabolism is, the more ATP the cells can generate. So the more glucose that's taken up, for instance, the more ATP can be generated. And it turns out that the cells that have a higher excess production of ATP are actually more drug resistant. So they seem to have more of an energy reservoir and ability to deal with stress. So I thought that was kind of a satisfying paper, actually, given all the talk about cancer cell metabolism and so forth, that this gave some structure to that. Sorry, I had to run through that a little too quickly. Now I want to spend my remaining 20 minutes on 12 minutes on talking about distal causation. So this is biology. We have now been building adaptive laboratory evolution machines that allow us to do evolutions in the lab under controlled conditions on scale. We generated hundreds and hundreds of endpoints under well-defined conditions. And here we are varying the objective function and finding out how cells satisfy objective functions in the most optimal way. And the results from this are just absolutely fascinating. So I'm just going to give you one example of that. Let me first show you Adam Feist and his machine. So this is the manual process as implemented and much hated by practitioners in the mid-2000s, going in every day, multiple times a day, passing cells reproducibly and so forth. This is the modern way of doing it, a highly automated platform that can pass 7 times a day that no human can, do 10 replicate cultures or more. Oops, there it is. Run by MATLAB. You can log in online and see what the machine is doing. And as Adam says, this is the new you. Dozens of pairs of hands doing it at the same time. So here is Douglas McCloskey that wanted to study the distal response to the loss of a gene product. We like to knock out genes and say they do this or they do that. They're essential, they do this and they do that. We never give the organism a chance to respond to the loss of the gene product and evolve it. So he did this experiment shown here. As Aidan mentioned, not all wild-type cells are optimal in the sense that we like them to be, to grow as fast as possible. So before we did this experiment, we first evolved it to our optimality, to as fast growth rate as possible. So we had a reference state we understood. Then we knocked the gene out and crippled the cell, and we had an unevolved knockout, which we then subsequently evolved. And then we looked at the endpoints. And you can answer a lot of questions, what happened at the systems level, because we got a lot of all these data types, what happens mechanistically, and what are the principles underlying the recovery from a loss of a gene product. Now, remind you, this is a very non-natural perturbation to a cell. It's only recently in evolutionary history that a high-end primate comes along and rips a gene out of a bacterial genome and says, what are you going to do now? But we did that. So here's the experiment. Pre-evolution to optimal growth rate that is predictive and interpretable by FBA. Then you look at your metabolic map and you select target genes to remove. Then you remove them and you remove them and do the ALE experiments. Here there are 8 replicates, 4, and so forth. So this is PGI that we may talk about, this isomerase here, where the cell is almost crippled when it's removed, but it recovers quite nicely if you let it evolve and adapt. Some of them you take out and the growth rate barely changes, but still its metabolically rewires itself. So there are subtle changes needed in the metabolic map in this case, and the cells do that. Now, so here's the way the data looks. This is interesting. So we start with big data analytics, if you will, PCA. And here's the metabolome for this knockout over here. So here's the start point. I forget now how many measurements we had, some dozens. I don't think there were over 100 metabolites measured. And so this is the wild type strain. This is the unevolved knockout. And you can see it really moves here on this PC1. So this this component seems to be related to systems optimality. So it's knocked off here to a very low growing state. Then it evolves, and you see it recovers on principal component number 1. But there's a component number 2 here that shows that these endpoints are different. So there are different evolutionary solutions to this challenge, but they are roughly equivalent because they end up roughly at the same growth rate. And this seems to happen over and over again, that the response to an evolutionary challenge has multiple outcomes. They may not be strictly equivalent, but they're very similar. And now we are studying the trade-offs. You know, what does this mode represent? Why are these different? They become the same on mode 1, which is the basic growth process, but how they get there is different. So this is the metabolome for one of our endpoints. Here is the RNA-seq, here's the fluxome, and here are all the different knockouts we did. And not in all, but in 3/4 of the cases, this pattern here is displayed. You're thrown into an optimal state, you recover from it, and you recover in different ways. It's not a uniform principle, but it's a prevalent principle. Now, I knew I would run out of time, so I did not put in all the other data here that shows you how you analyze this further, but I'll just tell you what the result is. This is a good story for those that love metabolism and the importance of metabolite levels. So here's what happens when we trace out this causality, and we're talking about distal causation now. So the first thing is you knock out the gene, like when you take out PGI, you disable ocular glycolysis. That's a big disruption, right? So the pathways change, the pathway uses change, the whole flux map changes. And that perturbs the concentration of all the metabolites across the network. Now, all the major transcription factors in E. coli sense a metabolite. CRP sensing cyclic AMP, CRA senses 1,6-bis fructose bisphosphate, and so on. So the transcription factors are sensing the metabolome. And so it's changing, and therefore the transcriptional program is completely screwed up. so the transcriptome becomes very suboptimal, but survivable. And when you look at that, you actually see that without the gene, I'm talking about homeostasis now, these metabolites thrown out of the homeostatic level, all the built-in regulations have to conflict with one another. So the system isn't wired to deal with that state. And so it has to be deconflicted. And what happens then with time is that the mutations rewire those regulatory circuits. They deconflict them and build up a new regulatory structure, or slightly, it's not that new, it's slightly altered, that builds up a new optimal homeostatic state, a new steady state at which you can get the best possible performance. And so we are now studying the set of mutations and how this rewiring happens, and it's giving us a granularity or an understanding of how the TRN, the transcriptional regulatory network, is actually wired in E. coli. So we're hoping to have that for you before too long. We've done hundreds of these evolutionary endpoints. We have a database now of 20,000 causal mutations. The way we do the passage and the passage volume is done in such a way that there's just a reasonable probability for causal mutations to pass into a new batch. So the mutations that fix tend not to be hitchhikers. We can get out the causal mutations for each case. Now we have a database of 20,000 mutations, 20,000 perturbations of the E. coli NG1655 genome that are all causal. They all produce a phenotype that is advantageous in some way. Can you imagine the value of that data set and how much biology is going to tell us, and actually tell us even more about the dimensionality of the genome and how it actually adjusts. So I think we are just opening up an era of figuring out how a genome takes it from one homeostatic state and to another one, either in response to a biological perturbation, chemical perturbation, or a physical perturbation. And we are pushing on all of these, by the way. And see, you're getting restless, but I am done. This, by the way, this ability to study distal causation like this on scale with respect to these perturbations, resequencing, getting the mutations, and we can, of course, genome edit to put them in there and assess causality, or even the way we do the evolution, we can pick clones that tend to be pristine because of the way the selection is done, and compare them, mutation by mutation. This is just astonishing. Real tests of FBA predictions, right? Okay, so we are learning how to read genome sequences better and better. That is leading to genome-scale science, and a big part of that is to take what we read out of these genomes, coupled to the bibliome, as we call it, all the literature data on the biochemical properties of gene products, and build knowledge bases, build these reconstructions. They become the basis then to study omics data sets, study physiology, going from components to physiology, like with the red cell and some of the other examples I gave. Metabolomics can be mapped onto reconstructed networks and analyzed, and they, since we like to, the metabolism, right? The metabolome becomes the most important thing, but actually, at least in microbial life, the metabolites are probably the key variables. And we can then integrate that data type in conjunction with other data types through this knowledge base using assistance biology analytics. And we now have a growing number of examples that shows us how we can rationalize, how we can understand phenotypes based on those analytic methods and those data types. So we at least have scientific models now that give us understanding, and occasionally we get predictive models that have proven to be useful for design, like strain design, to design new phenotypes that are desirable from a bioprocessing standpoint. I think I showed pictures and names of most of the people that did the work along the way, so I'm not going to spend too much time on this. And acknowledge our funding sources, which have been the NIH, the DOE, and now increasingly more the Novo Nordisk Foundation that was mentioned in the introduction. And I will come to an end and stop talking. Thank you for your attention, and I'll take any questions you might have.
Thank you very much for the inspiring and interesting talks. I'm pretty sure there are some, even though we are close to lunch, I think we have time for a few questions or comments.
Yes, E. coli, you mentioned more than 1,000 species genome sequence. We know a lot about the biology, and the models are improving. And still today there are about 35-40% unknown.
Yes.
What do you think, is there any general assumption about their roles and how to understand what they are?
Yeah, so this is just a great fundamental question. So, what's for lunch? How long should the answer be? So there are some. So there are about 1,400 of these in NG1655. About half of them have some annotation, like a membrane protein, likely chaperone, ATP binding motif, and the like. But the other half, there's like nothing. And those turn out to be the genes that are not expressed often under laboratory conditions. If you expose the strain to other bacteria or spec media or something like that, they come up. So they're probably signaling genes, and genes that will be important when it comes to studying microbial community, so we would really like to understand what they are. So we have started officially a program called the YOME, all the Y genes in their totality. We have a collaboration now with IBM and Watson, which is going to try to go into the entire literature through language processing and try to dig up everything that can be automatically generated about these genes and somewhat curated text. We are studying their expression. We now do multi-strain comparisons to see how these Y genes are used in E. coli strains of different lifestyles. So we've done that. So we are looking at that in detail as well. And so these are kind of, I don't know, the big data analytics approaches to this. We're also doing a bottom-up approach on selected set of genes. We, for instance, have predicted based on structural features of the protein that are made, whether they are transcription factors or not. And we are tagging them and doing chip exo experiments. And we're doing this for about 60 genes, and we have through about 30 of them. We've already found 20 transcription factors in this way. Some of them are reasonably broad acting, and some of them are responding to stresses that the cells typically don't encounter under laboratory conditions. So that's a bottom-up approach with a defined set of genes. We're also very, very interested in the transporters. And we can see in our transcriptome when we decompose it with ICA, not PCA, so independent component analysis, which decomposes the data set much better than PCA. You're seeing that some Y genes on the components are moving opposite to biosynthetic genes, so those are mostly likely transporters for that product that is made by that pathway. We have this for histidine and most of the amino acids now. And so we're going to go into a transporter discovery process through this analysis. So we're trying to close this gap systematically. It's an exciting process ahead of us, but at least we can take on a large-scale discovery effort here in a very structured way. And so let's see how successful we will be. We'll probably get, a fair chunk of that YO defined, but then there are always these CRISPRs, these genes with functions that you can't guess ahead of them, and who knows how many exciting functions are actually in those YOs. There undoubtedly are many.
Yes, we have time for the last question.
Just a follow-up question. How many of these genes are corrected? How many are wrong? You rely on literature. How much has to be rewritten in the biology?
So this is another great question. What's for lunch again? No, this is really a great question because it turns out many genes are multifunctional. And so you have one function annotated, but there's another function that you don't know, and you're never going to look for it in that gene because it's already functionally annotated. So many underground metabolic activities, for instance, fall into this category. So even if you have annotation to a gene, those 3,000 genes, there's always possible there are other functions in there. Often, it has happened that those are responsible for false predictions of the model. So they may be helpful in finding this out. How many other false assignments are to open reading frames in E. coli? The 3,000 that we're mostly focused on have a lot of bibliomic data on them, so I think that's unlikely. But the half of the YO that has some annotation, that annotation can be very flimsy and incorrect. So I just don't know how that's going to map out, but we should be aware of that. Try to figure out, all these secondary functions of every given problem.
Okay, I think we should continue this discussion during lunch. We have a small gift from Digital Life here. So, please.
Thank you so much. It's physical.
Yes, this is physical.